This protocol allows for the real-time assessment of various processes and biochemical parameters within the lumen of individual macropinosomes. These methods can be employed to drive discovery in the cell biology of macropinocytosis. This protocol provides the advantage of revealing heterogeneity at both the cellular and organellar level.
One day before the assay, seed raw 264.7 cells at a density of 5 × 10 to the 4th cells in 100 microliters per well in a glass bottom 96 well plate with black sides. On the day of the assay, check whether the wells are at least 70% confluent. Then wash all the wells with 100 microliters of HBSS pre-warmed at 37 degrees Celsius. Next, add 100 microliters of HBSS containing 0.025 milligrams per milliliter of TMR labeled 70 kilodalton dextran and 0.025 milligrams per milliliter of fluorescein labeled 70 kilodalton dextran in each well. Then incubate the cells at 37 degrees Celsius for 15 minutes. After the incubation is complete, remove the plate from the incubator and wash the cells 6 times with 100 microliters of HBSS. After the last wash, add 100 microliters of pre-warmed HBSS to the cells and place the plate on a microscope with a heated stage and chamber. After adjusting the excitation and emission parameters for TMR and fluorescein as needed, acquire an image from each well, alternating between the 2 fluorophores in between each well. After the first acquisition, check whether all the wells remained in focus across the entire plate